Good morning, children. Students, in the previous video, we were discussing about the Human Genome Project. Now today, we will complete that Human Genome Project. So today we have to discuss with the salient feature of HGP. Salient features of Human Genome Project. Okay? Now see, after studying everything, that what was our estimation, that it will take 13 years to complete the study about the human genome and all, complete sequence of the human genome, that will take 13 years and all. This thing we have already studied. Then we have seen some methodology, how to do and how not to do. We have seen everything. Okay, now we'll see some salient features. That is, point number 1 is the HGP contains 3.1 billion nucleotide base pairs. Okay? Next, we saw that average gene contains about 3000 base pairs. Okay, but the largest, point number 3, the largest human gene contains 2.4 million bases. Okay, and the largest gene is dystrophin, PRO. This is the largest gene. And next point, the total number of gene was 30,000. Okay, which is much smaller than the 3 trillion. Earlier, they were thinking that there are near about 3 trillions of genes, but actually they found that there are total 30,000, and these are present on these 30,000 genes. Okay, now next is the function of 50% gene. Okay, is still not known, is still unknown. Okay, that's why we study about the coding and the non-coding, and only we get about the 50% result. Clear? We do not know anything about the non-coding genes. So function of the 50% gene is still unknown. Next portion, next point, repeated sequence make up a large portion. This is important, children. Repeated sequence make a large portion. Okay, this means that the nucleotide which is present on the gene, it can be repeated. Okay, so this is important. Repeated sequence, this means that in our genome, there are set of nucleotides which are repeated. Okay, in our genome, in our sequence of gene, there are the nucleotides which can be repeated. So this repeated sequence makes the large portion in the human genome. Okay, next, less than 2.1%, less than 2% genome, they code for protein, and rest do not code for anything. So this is our 7th feature, that is only 2% genome code for protein, and rest do not code for anything. these are the points. Now, one more. Chromosome number 1 has maximum gene. This is our 8th point. Chromosome 1 has maximum gene. And what is the number of the gene? 2958 gene. Okay? And this is the reason, children, that whatever is the result, I'll tell you. 9th point. Chromosome Y, which is a male chromosome, Y has minimum gene. And what is the minimum number? It contains 231 genes. Okay? Next, 10th point. At 1.4 million locations. What is there? We find SNP. Okay? That is, full form is single nucleotide polymorphism. Okay? Now see the meaning of SNP. What we found? We found that at 1.4 million location, there can be SNP, sorry. So, see children, what is this SNP? Suppose I am drawing one segment of a gene. What it will be? AATTCCGG. Understood? Now, I told you that these genes are repeated. Most of the genes, they are repeated one. Now, suppose there is one more A. Instead of this A, if there is a blank, okay, then TTCCGG. Okay, then there is nothing. And this is called as single nucleotide polymorphism, SNP. A slight difference, a point difference in the composition of the gene is called as the SNP. So, these type of differences are found at 1.4 million places in the gene sequence. And this was the most important part, children, that by finding out these SNPs, we can easily detect the genetic disorders. Okay, that we have already studied what happens in case of point mutation. There is only one change at one point. And that point, that changes are present in the different places. And these different places are 1.4 million places. Clear? So, this is a SNP. One more, we can say that, suppose AATTCCGG. Okay, what we can draw? ACTTCCGG. Change? Okay, this is called as the SNP. So, what type of, when the sequence is changed at one point, when the sequence of the gene is changed at one point, then that is called as the SNP. And there are 1.4 million locations where we can find out these things. Now, last point, 11th point is, it was said that it will take 13 years to complete the project. But it completed in 2006. Okay. So it took near about 16 years to complete. And why it took 16 years to complete? Because the scientists, they had complete their work on all the chromosomes. And at last, they were working on chromosome number 1. And we all know that the chromosome number 1 contains 2958 genes. And this alone, this single gene took 3 years to complete their project. So they took 3 years to study on the chromosome number 1, and it took 3 years, so that the whole work was completed in 2006. Although it was decided that they will complete the whole work until 2003, that is within the period of 13 years. So all these are the salient features of the HEP. Now, what is the application of this? Why they have worked so hard? Why they have spent a lot of money? Why the whole world was involved in studying the Human Genome Project? What was the purpose of this? Okay, point 1. The first important purpose, to diagnose the disease. Okay, so what is the application? Application of HEP. Number 1, to diagnose the genetic disorder. Okay, very easily we can find out the genetic disorder, because we have already seen the SNPs. If there is a slight difference at one position, then that can be corrected. It will be first identified, taken out, removed. Okay, that we will see in the biotechnology how to do all these things. It will be removed and new piece of set will be set at that place. So we can cure the disease, the genetic disorders. So to diagnose the genetic disorder and treat it, not only diagnosing, and treat it. Okay, so this was the first application. Then what to do? We can treat it by using rDNA technology. This I will tell you in biotechnology, children. Recombinant DNA technology. The next point is, okay, by using this, the genome of non-humans organism can also be sequenced. Okay, so our first work was on HEP. Then the scientist worked on mouse genome project, which was the second largest. Then they worked on rice genome project. This was the third. Okay, then yeast and all. So by studying the Human Genome Project, we can work on all the other non-living organisms. Clear? Like Drosophila also. Okay. What is the next application? In the field of agriculture. This also we will see in detail when we will study about the application of biotechnology, children. We will see that we can form the flavor saver tomatoes. We can work for the rice. Okay, that is called as the rice fortified pot. All these terms we have to see, that we will see in the next chapter where we will study the biotechnology. So in the field of agriculture, we can use or we can diagnose. Clear? The next is, we can obtain different products. Okay, that is, we can obtain energy products by using this Human Genome Project. Clear? So children, this is all about Human Genome Project. Okay, now we have to see about the rice genome project, because that is also the first project done in the field of the plant. Okay, and why we have selected the rice? Because it is the #1 crop all over the world. It has a specific chromosome number. Okay, and the scientists have seen that all 90% of the chromosome, even 99% of the chromosomes, they are already genes, they are already well sequenced. So that we will see in case of rice genome project in the next part. Okay, till then children, you please prepare the notes of Human Genome Project as well as the DNA fingerprinting. Okay. Now in the next part we will see the next portion. Thank you.